now. Everybody should be on mute. I think you are. Obviously, unmute yourself if you have any questions. Let's see here. If you're online, you should now be looking at the PowerPoint. But before we get started, I'll remind anybody watching this, if I haven't told you else, if I haven't already told you so, go ahead and email me for attendance today, especially if you're watching the recorded version of this. Again, I'll remind you, we don't have lab today because lab from last week was extended to this week, so we're not meeting online. And that also got me thinking, next week's lab that will be online now, that was supposed to be in person, the way I'm doing it is I'm doing the experiments and I'm recording myself doing it, and I'm embedding that into the lab worksheet. And then I got to thinking, the reason I'm doing that is to keep you guys safe and myself so we don't have so much contact. But that being said, you know, you guys have paid for an education. So if ever we do an experiment, you know, we do a lab and then afterwards you're like, oh, I really like looking at the virtual lab. Now I want to do it myself. You've paid for that. So if that's what you want, contact me and we'll find a time to do it. I'll make some arrangements and it'll just be the two of us and we will do the experiment together. So keep that in mind. I don't want to because again, social distancing. And to be honest with you, I think what I'm doing online might be better than in person, because when you're doing these experiments in person, you're so busy like following the exact directions. Like, all right, put this into this tube, now put this into that tube, and write down what you see. Some people get so caught up in the details that they miss the big picture. Actually, I would say more often than not, they miss the big picture because they're so focused on the details. So with me doing the physical experiment, and then you just like using writing your hypotheses, and then looking at my results of the experiment to see if your hypothesis was correct or not. I don't know. I think there's advantages to that. But anyway, like I said, if you really want to do these experiments after we've done a lab and you're like, oh, I would really like to do that, then let me know and we'll make it happen. So any questions on that before we get started? Okay. Also a good time to remind everybody, independent work. Some people have done zero independent work. I'm telling you, you need to start now. It'll bite you in the rear if you wait. So you should really go ahead and get started. But anyway, let's get back to chapter 3. So we finished one half of the chapter basically, because the first half, we could say we talked about carbs and lipids, right? So carbohydrates, fats, steroids, things like that. And in my opinion, what we're about to cover, proteins and nucleic acids, I think these are much more important in the scope of this semester in this class. Because even though you're getting introduced to proteins now and you're getting introduced to nucleic acids now, but they're going to be concepts that we're going to talk about almost throughout the semester. So what we're about to jump into, in my opinion, is the most important in the context of this course. So here we go. Let's talk about proteins first. Come on, if I can get it to work. There we go. Like we've been saying throughout the chapter, one of the common themes, one of the things you need to look out throughout the chapter is the idea of the monomer versus the polymer, right? So with the carbohydrates, the monomer was the monosaccharide and the polymer was the polysaccharide. With lipids, it wasn't exactly a monomer polymer situation, but for example, the triglyceride was made up of the one glycerol and the 3 fatty acids, right? So you need to know the parts. Same with proteins. So the first thing you need to know about them is that they are polymers of amino acids. So if you're taking notes, definitely write that down. That will definitely be on the exam. It's a very important concept when we get into this stuff later, when we talk about how DNA works and how genetics works. So a very important concept. Proteins are made of amino acids. And then later, we'll even talk about how the amino acid itself, what it's made of. But for now, just know that proteins are polymers of amino acids. So you now know what the amino or the protein monomer is. And later, I'll tell you the technical name of the polymer. But for now, just know that amino acid, that's the monomer. Your book points out that over 50% of the dry weight of your cell is made of proteins. And that's important because remember, I keep saying it, and you do need to know it. Your cells are mostly water, right? But if you were to take away all that water, it's mostly proteins. And that should tell you just by sheer volume how important they are, right? The fact that you have so much, that means something. Specifically, they're needed in almost everything cells do. That's not going to be a test question. That's just more like, hey, why are we learning this? Why do proteins get a whole section in a chapter in the semester? That's why. And you'll see why later, how proteins are involved in almost everything a cell does. And your book also says that they are the most sophisticated molecule in the body. And in my opinion, that is more of a subjective statement than objective, but whatever. Excuse me. And I'm certainly not going to ask you that. So for the exam, here's your two important bullet points. The first two. They are polymers of amino acids. Actually, you know what? Just the top one. That's the only one. You don't even need to know this over 50% stat. That's just like implying, again, this is why it's so important. Obviously, there's a lot of them, so they must be important. So any questions on this slide? All right. This is decent information. I'm not going to ask you about it, but your book does point out that there are different types of proteins. So usually I think when people think of protein, at least outside of biology, they're usually probably thinking of this, right? Protein like muscles, either because you're building muscles like this person here, or from that picture at the beginning of the chapter, meat, right? Because you eat meat for protein. Well, meat is muscle, right? But there's also other stuff. You've got structural protein, so the stuff that makes up your fingernails, the stuff that makes up your hair, the stuff that makes up the horns right there, that's structural protein. That's just some examples. You have storage proteins, so seeds and eggs, which makes sense, right? Because they're storing things because you're going to grow a new organism from that. So it makes sense that you would need some storage protein. Transport proteins, that's actually really important in different biology classes. We would talk a lot about that. We will talk a little bit about transport proteins in this class, but not a lot. But let's just say they're really important. And then finally, enzymes. And in my opinion, as far as this class is concerned, enzymes are the most important, which is funny because they show a picture of a bottle because you can use enzymes, like, buy them at the store and use them. But when I'm saying enzymes are important, I mean in your own cells. And like I said, we're going to talk a lot about enzymes starting, well, obviously this chapter, but and then also in the next chapter. So enzymes are very important. But again, if you're taking notes or you're wondering what to study, you do not need to remember all these different types of proteins. The point is there's a bunch of different types of proteins. So any questions about that slide? All right. So like I said earlier, the monomer of a protein is the amino acid. And I told you later that we would learn about how the amino acid is built, like what it's made of. So that's what we're about to do. And you should know that there are 20 amino acids in your cells. In every living thing, there's 20 different types of amino acids. I might ask that on this question, but I'll definitely ask this later in the semester when we start talking about proteins again. So this is your introduction, and we'll come back. So each of these 20 amino acids, no matter which version it is, it has the same 4 parts, sort of, and you'll see what I mean. There's always the central carbon. There's an amino group. There's a hydrogen. There's a carboxyl group. And then there's the side chain. And the side chain is the part that's different. So every amino acid is going to have the same exact amino group, the same exact carbon, same exact hydrogen, the same exact carboxyl group, but their side chain will be different. Each of the 20 amino acids has their own different version of the side chain. And if you're taking notes, there's no need to write that down now because we'll talk about it later. And also, so you know, because this is a 100 level class and you guys aren't biology majors, I'm not going to require you to know all these different parts. What I might do is say, you have this molecule that has a central carbon, an amino group, a hydrogen, a carboxyl group, and a side chain. What is it? Does that make sense? So I'm not going to say, what are amino acids made of? But I might do the opposite where I say, you've got all these different things, and what is it? And then hopefully you'll remember that it's an amino acid. So again, it's not that you need to memorize all the different parts. But if I give you all the parts together, you should say, oh, yeah, that's an amino acid. So are there any questions about this page? All right, moving forward. The stuff on this page is what I've already told you. You just saw it in the picture, right? You've got the 3 common parts. No matter what amino acid, you have the same parts. The carboxyl group, the amino group, the hydrogen atom, and obviously the central carbon. And again, like I already said in the last slide, the variable component, the part that is different for each amino acid, that is the side chain. Actually, and before we move forward, let me move back one more time. This is the slide we were on earlier. Like I said, for all living things, there are 20 amino acids. So no matter what cell you're looking at, if it's your cell or a bacteria cell or a monkey cell or a banana cell, whatever it is, and you were looking at all the different amino acids, it's the same 20, no matter what. So that's one of the interesting things about life, right? We all have the same 20 amino acids. But the reason I put a star on there is because there's actually many, many, many more amino acids in nature, just not necessarily universal to all life. So that is a good independent work topic if you will look into it. What are the other amino acids? Where do you find them? Things like that. But anyway, moving forward, here's another picture. You've already seen it. This is just a different version. Again, central carbon, amino group, hydrogen, carboxyl group, and the side chain. Like I keep saying, we're kind of beating a dead horse here. Each different amino acid, each of the 20 amino acids, has its own unique side chain, and that gives it its special chemical properties. And we'll talk about that on the next slide when I can kind of show you 2 examples of them. And the side chain kind of defines the amino acid, which makes sense, right? Because all the other parts are the same. It's only the side chain that is different. So that's what defines it. None of that that's on the slide will be on the exam. But here's a good example. Here's 2 different amino acids, leucine and serine. And again, you can see the parts that are the same, the amino group, carbon, carboxyl group, hydrogen, all the same, right? It's the side chain that's different. And again, like I keep saying, none of you are chemists, none of you are biologists. So I don't expect you to know all the biology and chemistry stuff. But even without knowing the details, you can just look at the size and shape of this versus the size and shape of that and say, hey, those things have a different shape. Therefore, they must have a different function. And that is exactly true. This thing is going to fit into different places than this thing, right? Because even though we look at this as 2-dimensional stuff, it really is 3-dimensional. And these really are molecules that interact with each other and you repel each other and attract each other. So you can tell just by looking at the shape that these things are going to be different. And then I like that your book points out that this is hydrophilic and this is hydrophobic. So if you remember, hydrophilic things attract other hydrophilic things, but they repel hydrophobic things. And hydrophobic things attract other hydrophobic things, but they repel hydrophilic things. So these 2 things, if they were to meet, would repel each other, right? And if this hydrophilic side chain were to come in contact with water, for example, it would be attracted to it. If this thing, the hydrophobic side chain, came into contact with fat, like the stuff that makes up your cell membranes, it would be attracted to it. So again, the point here is we have 2 of the 20 amino acids, just 2 simple examples, and you can see how different they are, even without being an expert, just with this basic information. The shape, the size, and the fact that one's hydrophilic and one's hydrophobic. And it actually gets more complicated than that. We just don't get into it. These are just examples, by the way. There'll be no questions about leucine or serine. I'm not going to say, is leucine hydrophobic or hydrophilic, right? There's nothing to memorize there. It's just an example. So any questions on that slide? All right. Keep moving. Here's a big one. We're going to talk about structure function, as we often do in the semester. Your book points out that cells kind of put these amino acids together, which is obvious, right? They don't just spontaneously come together. And again, amino acids are the monomers, so you're going to have to put them together to make the polymer. But your book does point out that when you put these amino acids together, that's called a peptide bond. So you might need to know that. If I ask you that, it will not be in this chapter, but it'll be later in the semester. So we will come back to this idea of peptide bonds. But if you recall, this is the first time we've given it a specific name. So far, the only thing we've said in this chapter is if you take two monomers together and put them together, or if you take them and put them together, that's called a dehydration reaction. This is still a dehydration reaction, but it's such an important reaction, they gave it its own name. It's a peptide bond, right? That's what keeps them together. And I think the only reason your book really points it out is because the next word is an important word that you need to know, which is polypeptides. So you need to know that word. So you might get a question where it says, what is the monomer of a protein? And the answer is amino acid. And then you would get, what is the polymer of a protein? And the answer is polypeptide. Hope that makes sense for everybody. Now you know the monomer and the polymer. Let me scoot over here real quick. See something. Oh yeah, that'll work. So your book points out then that a functional protein is one that has one or more chains of these polypeptides that have been twisted and folded and coiled into a molecule of a unique shape. And I'm about to differentiate here because some people get this a little bit confused. On one hand, we're saying the monomer is the amino acid and the polymer is the polypeptide. So then some people might say, well, then what's the difference between a polypeptide and a protein, right? They sound like the same thing, but they're not exactly the same thing. And I'll show you why here in a second. If you're in person, you can see this. I don't know if you're online, if you can see this, but for those of you who are online, can you see what I'm holding up? Can someone tell me what that is? Maybe you can't see it.
Paper airplane?
Yeah, it's a paper airplane. Would you agree in person? Yeah, it's a paper airplane, right? But what else is it when you think about it? I mean, yeah, technically it's a paper airplane, but someone could also say it's a piece of paper, right? And that's kind of like the relationship between a polypeptide and a protein. A polypeptide is just a piece of paper, so to speak. It's not a protein until you've taken it and it's been folded in its correct form. Does that make sense? I hope it makes sense. So polypeptide is just the string of amino acids. It's not the protein until you've shaped it into the right shape. And that's why I included this picture down here. I don't know what it is. I don't know if it's a scarf or a little blanket, but whatever it is, it's something that's knitted, right? So the same kind of relationship here. Let's call it a scarf, right? You can say, oh, that's a scarf. But also technically, it is a piece of yarn, right? So the yarn would be the polypeptide and the scarf would be the protein. Because the yarn is just a piece of yarn until it's been twisted and folded into the right shape. At that point, it is now a scarf. So does anybody have any questions about the relationship between the word polypeptide and protein? Okay. And it's not that I'm going to ask you the difference. As far as exams are concerned, those words are kind of going to be interchangeable. But I just want you to know in your head there is technically a difference between a polypeptide and a protein. Even though they're the same thing, they're made up of the same amino acids. It's not until it's folded that it becomes a protein. Anyway, if there's no questions about that, the next slide is going to show you something that I've already talked about, which was the peptide bonds. Excuse me. Again, here you have a monomer and you have another monomer. When you put them together, it's a dehydration reaction. And it's such an important thing for proteins that your book went ahead and told you they're telling you what the bond is called, and it's called a peptide bond. And again, I think normally I don't think you would need to get into that kind of detail, but hopefully the fact that that's called a peptide bond will help you remember that they are called polypeptides. So any questions on that slide? All right. So your book, excuse me, I won't even say that. Your cells, like in each one of your cells, there are thousands of different types of proteins, but we only have 20 different amino acids. So then the question is, how do we do that? And before we even get there, let me point this out. That's a decent decent independent work question. So I'm saying that there's thousands of proteins, but if you want to look it up, exactly how many proteins are there in the human cell? So you can look that up if you want. But anyway, for this class, we're just saying that there's thousands. Not that that's going to be a question on the exam, but we just want you to. I just want to get you thinking, like, all right, how do we have so many different proteins when we only have 20 different amino acids? And your book uses a good analogy, and we're talking about letters, right? So the English alphabet has 26 letters, right? That's kind of close to 20 amino acids. But even though we only have the 26 letters, depending on how we put them together, we have a lot of words, right? Does anybody know how many words are in the English language? Not that you need to. But as of 2016, there were 1,025,109 words. So you can see, not that you need to know that. The point here is that from 26 letters, we were able to make that many words. Plus, there's rules in the English language, right? You can't put this letter before that letter, and this letter always has to come after that letter. So even with all those rules, and the fact that our words really aren't that long, even with that, we were still able to get over 1 million words. So that's how we can have so many different proteins with only 20 different amino acids, which is good to keep in the back of your mind. And it's a good answer to a question that may have popped up in your head, but I won't ask about it on the exam. So any questions on that side? All right. If you download the PowerPoint, you can watch this video. It's all about protein structure, primary protein structure. If you were taking a higher level biology class, we would talk about the difference between primary and secondary and tertiary and quaternary, but we don't really get into it in this class. If you're interested in it, ask me or look it up for independent work. But again, we're talking about structure function. It's the amino acid sequence that determines the three-dimensional structure of the protein. So yeah, it is like, here's a great example. It's this amino acid and then that amino acid and that amino acid. And at the very basic level, you're just like, okay, what's it matter? It's just this one attached to that one, attached to this one, attached to that one. But the way they're attached together, right, the exact sequence, that actually determines what shape it is. And this is a very basic example. I found this on the internet because it only shows one loop, so I wanted to include it. So you can see this amino acid has a little sulfur coming off of it, sulfide, so does this one. Those things are attracted to each other. So the fact that this polypeptide has that amino acid right there and that amino acid right there, because of that, this thing has that shape, right? So I hope that makes sense. And it's just an example, so you don't ever need to memorize it. But the point here is the fact that those two amino acids were there gave this thing its three-dimensional shape. And that's a very simple explanation or example. It actually is usually a lot more complicated than that. So are there any, oh yeah, sorry. And your book also says that, so like this isn't just trivial, like, oh, who cares about the three-dimensional shape? The point here is that the three-dimensional structure enables proteins to carry out their specific functions. If you're taking notes, I would definitely highlight that or circle it or bold it or write it down, because that question will come up again and again. So and I'm trying to think of all the different ways I could ask it. Like a protein's function is most dependent on what? And the answer is its three-dimensional structure or shape, however you want to say it. And again, this would be multiple choice. So you might have something like a protein's function is most determined on what? I might have size or pH or weight. But the answer is three-dimensional structure / shape. Those can be interchanged. And this next one kind of explains why that's so important. It's because nearly all proteins work by recognizing and binding to some other molecule. So it's all about the shape. And again, all these molecules we've been talking about since chapter 2, I keep saying, even though you're not chemists, like, look at the shape of this thing. You can obviously tell that it's going to do something different than this because it has a different shape. And this is what I'm getting at. We're finally talking about it. It's because of all these proteins that have their own shape. And you'll see a lot of it next chapter when we talk about enzymes and how things fit like a lock and a key. But are there any questions on this slide? All right. And again, to me, this is the most important bullet point on the slide, that middle one. Protein's function is based on its shape. Here's another example. So the last slide I showed you gave you an example, and this is a book example. So here's this long chain of amino acids, right? That's not the shape of it. They just had to do that so it would fit into the slide. But you can see it's just a long chain of amino acids. For some reason, they zoomed in on this one, alanine. So that's just the chain of it. But if you look here, you can see how it folds and twists. So that's the shape of the protein. That's the shape of all these amino acids. This is just a polypeptide. These two, those are proteins. And they're both the same thing. It's just this is a different model than this, right? So again, it's all about the shape. So does anybody have any questions on that side? All right. Here's something very important, especially later in the semester. I might not ask much about it now. I'm just introducing it to you now, but later in the semester, this will be very important. But if you have a slight change in the sequence, right, the sequence of amino acids, and let me back up again, a slight change in which one of these is attached to which one of these, if you change it a little bit, it could be a really big bad thing. There's an example here. I'll tell you about it in a second. No, I'll just talk about it now. Let's just say you can ignore this picture because there's a great picture from your book that I'll talk about next. But your book uses hemoglobin as an example of how a slight change can be bad, and that's on this next slide. So here is the normal chain for hemoglobin, right? And this is what it normally looks like, and that's what a normal blood cell looks like. But here, we have one different amino acid. That's it, just one. The rest of them are exactly the same. If you change that one amino acid, now the protein is misshaped, right? The protein, one of the 4 proteins in that shell or a cell, that's misshaped. So now the whole cell is misshaped. And we'll talk about that disease later on, but sickle cell anemia, that's a pretty big disease, and it has a lot of negative consequences, all because you had that one change in amino acid. And again, I'm just introducing you to this now. That's going to be a really important concept later in the semester. For now, it's just an introduction. Are there any questions about that? All right. Actually, let me back up too. I'll go back up to this non-book picture. So again, this picture is not in your book, and it's really complicated, so don't feel like you need to memorize anything or truly understand it. The point here is you can see that this thing kind of like spirals, right? And the reason that's happening is because you have these hydrogen bonds, H to O, H to O, right? H to O. You can see all these different hydrogen bonds. So now imagine if you changed one of these amino acids to where there was not an oxygen or there was not a hydrogen, then you would lose the hydrogen bond, and then it wouldn't have that shape. Same thing here, a bunch of hydrogen bonds, right? So, and for the record, that's not exactly how it works because technically I'm telling you about amino acid parts, the non-side chain is responsible for this, but it's just to help you visualize that if you make a slight change that it could mess up the whole protein. All right, moving forward. This is basically what I've already said earlier, but now it's in writing. One difference in the amino acid sequence is enough to cause the protein itself to fold into a different shape. And again, like I keep saying, and you need to know this for the exam, a protein's function is based on its shape. Therefore, if you alter the shape, you alter the function, and a lot of times that can cause diseases. And your book lists some of them. You don't need to write these down because, again, we'll talk about these later in the semester. But some of them are things like prions, severe nervous system disorders, things like mad cow disease, things like that. Any questions on that side? Speaking of mad cow disease and prions, here's a great example. This is just an example. There'll be no questions about this on the exam. But it is a great example of how if you take a protein and change the shape of it, it causes a disease. So here you have a normal brain, right, with all these normal proteins in it. Then you have these diseases called prions, which themselves are just basically misfolded proteins. But they're contagious in that when that misfolded protein touches this normal protein, then it becomes misfolded. And then they touch another normal protein, and it becomes misfolded. So Polypeptide itself is still the same. The polypeptide, right? You still have the same sequence of amino acids, but this disease makes them fold the wrong way. And basically, you get your brain becomes spongy because you have all these different holes in it. And your book points out there's some examples, like I've already said. You've got mad cow disease, which is bovine, spongiform encephalopathy. Kuru, that's an interesting one if you want to look it up for independent work, because Kuru is basically only passed by cannibalism. So humans eating other human brains, that's how you get Kuru. And then fatal weight loss in deer, elk, and moose. I know one of you hunts. I think one of you hunts. I remember talking about that. So if you're into that, you could look up this disease for independent work, because there are like chronic wasting disease. There's this disease in deer in West Virginia. So that hits close to home. So any questions about this slide? And again, I probably won't ask you about prions in this chapter, this exam. I might ask you about it later. And even if I do, we're going to revisit this idea and you'll get the same information, and that'll be that. So if there's no questions, we'll move forward. Here's an important concept, very important later in the semester, but I'm introducing it to you now, and I want you to know it now. That way, later in the semester, I don't have to reteach this to you. This is an important concept. Again, a protein's shape is, its function, is based on its shape. Therefore, if you mess that up, you mess up the function. And your book points out that a protein's shape is sensitive to its environment. And you should know enough now from chapter 2, you should know enough basic chemistry where some of this will make sense. For example, pH, right? What do we say? What does it mean when I say something has a low pH? Like I said, that has a pH of 2. What does that mean? Yes, it means it's acidic. But in the terms of like H's or OH's, what does that mean when I say something has a low pH? That's something you should know. But what's that? Yeah, it has more hydrogen ions, right? It has more H pluses. So think about that. An H plus is obviously a positive charge, right? So if something's pH drops a lot, it's going to have a lot of H pluses. If the pH goes up a lot, you're going to have a lot of OH minuses. Those are charged things, right? Fully charged. If you look at this picture here you have something, those little dots, those are hydrogen bonds, right? So that's a partially negative oxygen attracted to a partially positive hydrogen, right there and there, right? That's what you're looking at there. And that's only partial. So imagine if you lower the pH and you introduce a bunch of fully positive hydrogen ions, those things are going to come in here, right? The oxygen is going to be attracted to that hydrogen ion because it's fully positive. It doesn't care about this partially positive hydrogen. And then likewise, if you were to raise the pH and introduce a bunch of OH minuses, obviously the hydrogen is going to be attracted to the fully negative hydroxide ion. So what I'm saying here is this thing is held together because it's in its perfect pH, whatever pH it might need. But once you start messing with it and adding a bunch of H pluses or adding a bunch of O minuses, you're going to break these bonds. And if you break those bonds, excuse me, that thing is going to unravel, right? And again, if you change the shape, you change the function. So that's why pH is such a big thing. Another one is temperature. We haven't really talked too much about that. How can I say this? So there's bonds holding these things together, right? There's those hydrogen bonds that we've already talked about. And the basic concept that you should know, not for the exam, just to understand what I'm talking about. Basically, and there's exceptions to this, but basically the chemical bonds can be broken if you give them enough energy. And hopefully you do remember when we talked about temperature, that is energy. Like when we say we increase the temperature, we're saying the molecules are moving around faster, we're increasing the energy level. So what happens is, if you were to raise the temperature on this, eventually these bonds would break because those molecules would have so much energy that they're no longer holding on to each other. And like we said in chapter 2, hydrogen bonds are the weakest of the bonds. So you give them enough energy and that whole protein's going to unravel. So any questions about this slide? I know that was long-winded. Basically, you need to know that a protein shape is sensitive to its environment. And again, its function is dependent on its shape. So if there's no questions for that, we'll move forward. Oh, yeah. Let's see, 8:34. We might be able to get through chapter 3 today. We'll see. So there's no questions on proteins. We'll move to nucleic acids. All right, there's two basic things nucleic acids do. They store information, and they provide instructions for building proteins. Yeah, never mind. I was about to really differentiate between those two, but we'll talk about that in chapter 10. There's two different types of nucleic acids, and you do need to know this for the exam. Know their abbreviations. You don't need to know the full name yet. Maybe later you will. There's DNA and there's RNA. And that should be easy to remember because of the NA part. The NA stands for nucleic acid. So hopefully you can remember that. Does anybody have any questions on this slide? If you're taking notes on this next slide, you don't need to write anything down because we're going to have a whole chapter devoted to this next slide. So I'm going to introduce it to you now and keep it in the back of your mind, but don't worry about studying it yet. Excuse me. So let me back up where we were. Like we said, nucleic acids provide instructions for building proteins, and that's all a gene is. Your book definition of a gene is, it is a unit of inheritance encoded in a specific stretch of DNA that programs the amino acid sequence of a polypeptide. An easier way of remembering that, especially for this chapter, a gene provides instructions on how to build a protein. That's it. So when you hear genes, I'm sure you've heard that word a lot. When people talk about genes, that's what they mean. It's just instructions on how to build a protein. Those instructions are written in a chemical code that have to be translated, right? So you have this chemical code of DNA, and that has to be transcribed into a chemical code of RNA, and that has to be translated into a chemical code of amino acids. But we will talk about that later in the semester. So again, if you're taking notes, for now, the only thing I think you really need to know is that a gene provides instructions on how to build a protein. And of course, that I do like that they mentioned that it's a unit of inheritance, meaning, yes, a gene provides instructions on how to build a protein, like I keep saying, but also because it's written in your DNA, it's also passed along. So, you know, you and I, there's only one person here, but you and I might have the same, well, we do. Let's say we have protein X. You have protein X, I have protein X, every human has protein X, but your gene might be written a little bit different. So when your body builds protein X, it looks one way. And then my gene might be written slightly different. So it's written another way. And then when I have kids, or as I have kids, and you do or will, you're going to pass that along. So their protein X will be written and built the same way. But again, for this exam, you don't need to worry too much about genes because we'll talk about them. We'll give them a whole chapter later. Technically a little bit more than one chapter, but is there any questions about this slide? Okay, here's a picture of everything I was saying. Imagine this is a long stretch of DNA. It keeps going around the room. You can't even see it. But we just zoom in on this one portion because that particular portion provides instructions on how to build a protein. And it's written in the DNA. And there's some machine, there's some cellular machine that comes in and attaches right there, and it moves down the strand and it reads the code and it builds a new molecule based on what that code says. That new molecule is RNA. And then some other machine comes and attaches to the RNA and it reads the code. And from there, it has the instructions and builds the protein, right? This amino acid attaches to this one, attaches to that one. And again, we'll talk about all that later in the semester. So any questions so far? All right. Again, the common theme of this chapter is monomer versus polymer. So again, as a reminder, the monomer of a carbohydrate is a monosaccharide and the polymer is a polysaccharide. The monomer of a protein was the amino acid and the polymer of the protein was the polypeptide. So now for nucleic acids, here we go. The monomers are called nucleotides, and you need to know that. And to me, I don't know, I always thought this would be one of the easier ones to remember. People always mess this up, but I don't know how. Because nucleotides, it sounds like nucleic acids. And you're only going to need to know the difference between the major molecules, right? So the question will be like, nucleotides are monomers for what? Obviously, it's not going to be carbohydrates. Obviously, it's not going to be proteins. Obviously, it's not going to be lipids, right? Nucleotides are the monomers of nucleic acids. There's 3 parts. I don't know if I'm going to ask you about this on the exam because, again, we're going to cover this in Chapter 10, but we'll at least talk about it. The 3 parts of a nucleotide are a 5-carbon sugar, a phosphate group, and a nitrogen-containing base. And I bolded the words, like if I were you and I were studying, because this is a 100 level class, you could just remember it as sugar, a phosphate, and a base. Yeah, it's 5 carbons, but that doesn't matter to you guys. Yeah, technically it's a phosphate group, but that doesn't matter too much. And yes, technically that base contains nitrogen, but that doesn't matter too much either. And here's a picture of what I was saying. There's the phosphate group, there's a 5-carbon sugar, and here's a base, a nitrogenous base. This one happens to be a cytosine. And you do not need to know that for the exam either. But again, the most important thing for you at this point in the semester is you know that the monomer for nucleic acids are nucleotides. Excuse me. So again, there's the picture, not from your book. The next slide shows a picture from your book. There's 2 different ways of looking at it. Again, phosphate group, 5-carbon sugar. There's your nucleotide, excuse me, your nitrogenous base. And this is the same thing. It's just drawn a different way. Phosphate group, 5-carbon sugar, and your nitrogenous base. So any questions on that? All right. So when we were talking about proteins, we said that they were made up of amino acids, right? And amino acids, there's 20 different varieties of them. And each amino acid, it's all the same, right? The central carbon and the hydrogen and the amino group, all that's the same. Only part that was different for each amino acid was the side chain, right? So it's similar with nucleic acids, and you do need to know this. DNA has 1 of 4 nitrogenous bases. So the same way amino acids, the same way there's 20 different versions of amino acids, there's 4 different versions of DNA. And for this exam, you could just memorize the letters A, G, T, and C. Now, when we really start talking about DNA later in the semester, then you might need to know the whole name. But for now, if anything, you just need to know A, G, T, and C. So again, I like to compare and contrast here. There's 20 different amino acids, right? Just the same way for DNA, there's 4 different DNA nucleotides. And with the amino acid, 3 parts were the same. The only part that was different was the side chain. For the DNA, it's the base, right? It's the base that's different. Actually, let me back up too, for just for the picture. So each DNA nucleotide is going to have the same phosphate group and the same sugar. This is the part that's different. It's the nitrogenous base that's different. Just like with the amino acid, it's the side chain that's different and everything else is the same. With DNA, it's the phosphate and the sugar that are the same, and it's the base that's different. So any questions about this slide? So there's a picture of A, G, T, and C. You definitely do not need to memorize you don't even need to memorize the name yet. Maybe memorize A, T, C, and G. You certainly don't need to recognize them by what they look like. I'm not going to give you a picture of this and say, is that A, T, C, or G? Right? You don't need to know that. So any questions? All right. So again, we're still talking about the monomers. We're talking about the nucleotides. So what kind of a reaction do you think it is that brings these 2 nucleotides together? And at this point, you should know, because this is the last portion of the chapter, and we've been saying it over and over in the chapter. We were bringing in 2 monomers, 2 nucleotides. So what do you think that might be called? What kind of reaction?
Dehydration.
Yay, there you go. Dehydration reaction. That's when we take nucleotides and we make polynucleotides, which is the next word you should know. So again, the monomer is the nucleotide, and the polymer is polynucleotide. So now you know that for nucleic acids. Your book points out that it's a covalent bond that brings them together. And that's something that's going to be really important later. So for now, just keep it in the back of your mind. You don't need to know it yet. Just keep it back there. That will be very important later, that it's covalent bonds keeping those things together. And later, it'll be very important that you know that it's a sugar phosphate backbone, meaning the sugar of one is attached to the phosphate of another, which is attached to the sugar of another, so on and so forth. The point here is it's the sugar and the phosphates that are covalently bonded to themselves. The bases, the A's, the T's, the C's, and the G's, they're kind of just hanging off. Those things are not covalently bonded. It's the sugars and the phosphates. And again, this concept will be really important later in the semester. For now, I'm just introducing it to you because your book introduced it to you. The only thing you need to know on this slide for this exam are that this word right here, polynucleotides. So the monomer is the nucleotide, the polymer is the polynucleotide. And yeah, technically you need to know it's a dehydration reaction, but I just keep including it because I'm just trying to beat that home. When you bring two monomers together, it's called a dehydration reaction. So are there any questions on this slide? Okay. This is going to be important later. For now, I'm just introducing it to you. DNA is double-stranded. And in this double-stranded molecule, the polynucleotides are kind of coiled around each other, which is why it has that twisted shape. And technically, that twisted shape is called a double helix. These are all things I'm going to teach you again later in the semester that you need to know. For now, I'm just introducing you. The bases on one strand of the DNA hydrogen bond to the bases on the other. Because again, remember, the sugar and the phosphate are covalently bonded to each other, and the bases are just hanging out doing their own thing. So they're bonded to each other with hydrogen bonds. And I don't know, I might ask you this on this exam. I'll definitely ask you later. But you definitely need to know eventually that A pairs up with T and G pairs up with C. So if I told you that, I don't know, we looked at the DNA of this cell and 30% of it was A, you could automatically deduce then that 30% would also be T, right? Because for every A, there's a T. And I probably won't ask you that on this exam, but I would definitely ask you later in the semester. Are there any questions on this slide? The next slide I'm going to show you is a visual of this slide. All right. So again, that's just a visual. There's your double helix. See, this is one strand that I'm pointing at. This is a completely different strand. Again, sugar, phosphate, sugar, phosphate. Those things are covalently bonded. And it's these bases that are just jutting out, hydrogen bonded to the other ones, and that's what holds the two strands together. So when you're looking at a DNA molecule that you always see, I'm sure you've seen this picture a lot, technically what you're looking at is two strands of DNA, right? It's not one strand, it's two that have been hydrogen bonded together. The book also points out, because so far we've been talking about only DNA, there are some differences between DNA and RNA. There's also some similarities. As far as the monomers and the polymers, that's still the same, in that the monomer is still the nucleotide and the polymer is still polynucleotide. The nucleotides still consist of a sugar, a phosphate, and a base. So those are the things that are the same. We haven't got into the differences yet. But are there any questions on this slide? All right. Here's the part you should know. You might get a question about this on this exam. You'll definitely get a question on this later. How is RNA different than DNA? The first one is that the sugars are different. For RNA, you have ribose, not deoxyribose. And that's the difference in the name, right? RNA versus DNA. The next one, there are no T's in RNA, right? So DNA has A, T, C, and G. RNA doesn't have that T. So instead of a T, it has a U. And the other big one is that RNA is a single-stranded, and DNA, as you already know from what we just talked about, is double-stranded. So are there any questions about this slide? I know I'm going through it pretty quickly, but we're running out of time. And it's a pretty simple concept. You just 3 things you need to remember. All right, if there's no questions on that slide, this next slide shows you, because again, there's no T in RNA, there's a U instead. Hey, and that's what the U looks like. So I mean, it almost looks identical, right? Unless you're studying this and you're a biologist or a chemist, I mean, that should look the same to you. But all right, when we get back, we're going to skip that. We get back, we will be done. Yeah, we did finish. Good timing. All right. So all that stuff I just skipped. As always, that's the stuff like the evolution connection that's in every chapter and the process of science that's in every chapter that I never test you on. So as you're reading it, if for some reason you do have questions about it, even though you don't need to know it, contact me and we can talk about it. But other than that, we are done with chapter 3. And then next week, we will decide when to take the exam. I can tell you it will not be on Monday because I want to give you some more time to study. So everybody check your emails this weekend because I will or check, Google Classroom because I'll be asking you when you want to take the exam. So, all right. I'm going to stop recording, and if you need me, I'll see you in office hours.